Okay, going on, now we're going to talk about different types of experiments. So, first thing you want to make sure you know how to differentiate is the difference between an observational study and an experiment. An observational study observes individuals and measures the variables of interest, but does not attempt to influence the responses. So, in an observational study, the researcher is observing something that is already happening. Okay? But we're not having an influence on the results. So, let's say I wanted to do experiment or observational study dealing with your heights, a study involving your heights in your AP Stats class. So an observational study is where I would just observe your heights, record everyone's heights, have everybody stand up at the board, measure their height, write it down. That would be an observational study because I'm the researcher. I'm not having an active role in influencing the response variable in the study. But another kind of example that would be an experiment is I told half of you guys to take this pill that I think is going to increase your height over the course of 2 months, and the other half class is taking a placebo. And then I record your before and after heights after that. That would be an experiment because I'm giving you something that's going to potentially influence the results. That pill that I'm giving you could potentially influence your ability to grow. So that is the main difference between an observational study and experiment. Really important that you know the difference between those two. And like it says here, when our goal is to understand that cause and effect experiments are the only source of fully convincing data. So anytime that you're doing a study, if you're doing a survey, observational study, you can establish correlation. So correlation is that if one variable goes up, it affects the other, okay? But we're not necessarily saying that it causes the other one to go up. So, for example, if we're dealing with a cause and effect relationship, we want to make sure that we're using an experiment to do that because we're controlling as many variables as possible. So if we're controlling as many variables as possible in a controlled experiment, we can have a stronger sense of saying that our variables have an effect on, cause and effect on each other, whereas in an observational study, we're not controlling all those variables. So there's a lot of different variables that could come into place. So an experiment is the only, controlled experiment is the only way that we could say and establish a cause and effect relationship. That does not mean that an observational study or survey is not useful, but if we want to have that pure cause and effect relationship, we need a controlled experiment. Next, we have confounding. So confounding occurs when we have two variables that are associated in such a way that their effects on the response variable cannot be distinguished from another. So that's basically where we have multiple variables that are changing, so we don't know what's influencing the results. So, for example, if I wanted to see our different balloon example, I don't know if you remember it all the way back in, I think it was September, we did an experiment to see what the relationship was between the length of the balloon and how far it flew in the air. But we didn't have good results because there was a confounding factor because the different balloons had different shapes. Some were circles, some were longer, some were small, some were big. So we weren't sure if the flight time was due to the different shape of the balloon or the length of the balloon. So if we did a better experiment with that, we want to keep the shape of the balloon the same, so then the only thing that we're changing is the length. So anytime we're having confounding variables, that's where we have multiple variables that could be influencing our results. We want to try and avoid that in an experiment. Now some terminology when we're designing our experiments. The specific condition applied to the individuals An experiment is called the treatment. So that's what you're giving the individuals, and that's where you're seeing if it has an effect on your response variable. So for example, if I was doing the experiment where I'm giving you the pill to see if that helps you get taller over the course of 2 months, the pill and the placebo, those would be the treatments that I give out randomly to each group. So a treatment is what the researcher assigns to the individuals in the study that they think is going to influence the results. Experimental units or individuals are the units that are getting the treatment in the study. So keep on using that same example with the height pill. The individuals, the experimental units would be each of you in class, and if the individuals' experimental units are human, they can also be called subjects. So here are some of the principles of experimental design. First, we want to be able to compare our results. So that means that we have multiple treatments, right? Because if I give everybody in the class the height pill, I don't have anything to compare it against. But if I have the height pill and then the other half the placebo, I have something to compare the height pill against. So we want to make sure you have comparison. Random assignment, we want to randomly assign the treatments just to eliminate bias. Remember, we can even have a double-blind experiment when the person administering the treatment doesn't even know what the assignment is, the treatment is, to help increase that bias. So random assignment is essential because we don't want people picking their own treatment because that could influence our results and give us bias in our experiment. Control, we want to try and control as many variables as possible in our experiment. Otherwise, there could be other variables that are affecting our results. So if I was doing that height example, I would want to make sure that all of you are having similar diets, similar amount of exercise, similar sleep schedules throughout those 2 months, because those are things that might affect your height. All right, so we want to make sure that you're controlling as many variables as possible, except for the treatment, so we can see if the treatment's having an effect or not. Replication, we want to make sure we're doing this experiment as many times as possible, because the more data we have, the better picture we have of what the population, the actual results are going to be. It's like if you're doing a survey, you'd rather have 100 people on your survey rather than 5. Then we have statistical significance. So anytime where they're talking about statistically significant, that's something in your brain you want to go and do a significance test. All right, we spent a lot of time talking about that already, so I'm not going to go through that in depth. Now we have 3 different types of experimental designs that you're going to see. First one is completely randomized. So that's where, say we start off with 14 people in the AP Stats class. I split them up into 2 simple groups: treatment group, the height pill; the second group, the placebo group, and I randomly assign 7 people into group the treatment group, the height pill, and then 7 people in the placebo. Record the results and compare. Then, so that is the most kind of like simple form of experimental design. Again, we want to make sure we have a control group. And anytime that you see a response to a placebo, remember that's called the placebo effect. So if we saw people's height increase just as with the placebo, then that would probably be a placebo effect. It's like a mental thing that they think they're getting taller, so maybe it's actually making them taller. And like I talked about before, a double-blind experiment, that's where the person administrating the treatment doesn't know what treatment they're giving out. So if I brought in another student and asked them to give out pills from container A to half of you and pills B to the other half, they wouldn't know which one's the treatment and which one's the placebo. So that's a way to help, again, reduce some possible bias. Next, we got block design. So that's where essentially it's completely randomized design, except we're splitting up into groups based off a variable that could influence the results. A common one, especially for like pills, medical treatments, is splitting people up by gender. So if I was doing this with a class of 20, and half of them were, actually, let's do a class of 40. Half of them were males, half of them were females. I would first, I would have 4 different groups that I'm randomly assigning treatments for. I would have 10 females get the height treatment, the actual height pill, 10 females that get the placebo. Then I'd have 10 males that would get the actual height treatment, 10 males that would get the placebo. So that way I can break it down and compare and see if there's a difference in results between the male and the female, between the treatment and the placebo. And then the last pair type of study is a matched pairs design. So this is a randomized blocked experiment in which each block consists of a matching pair of similar experimental units. So this is where we're essentially subtracting our end result to see if there's a difference between those two. And remember when we did our significance test, this was a matched pairs t-test. We were doing a significance test with that. So for example, with our height example, a matched pairs test, we want to do a before and after height, right? So we'd have 2 similar individuals, experimental units that we could compare. The before height and the after height for each person. So you could have a before and after for each individual, which would make it matched pairs. Or you can match up individuals that are similar with the different treatments. So if I was giving you like a pill that's supposed to increase your IQ and have you perform on different tests, maybe I'd pair the top 2 students with the top 2 GPAs and give one the pill and one the placebo, and then subtract the results, compare those results with each other, and then match up the next 2 highest GPAs all the way down until we get through everybody. So a matched pair is where you have either before and after for one individual, or you have 2 different data points from individuals that are similar, and then you're comparing, subtracting them. So I know there's a lot of vocab, and we're going to get some practice now going through 2 free response questions that you're going to send to me today. So just, again, if you need to, print off this study sheet. You memorize all these terms, you're going to be in really good shape. It's everything you really need to know just on this 2-page sheet. So really good resource, or 3 pages. So it's a really good resource for you to study with over the next 2 weeks.